All right, welcome. So you probably think of your DNA as this fixed, stable instruction manual for life, right? Like a book that never changes. But what if I told you that parts of that book are constantly ripping themselves out and pasting themselves onto new pages? Let's dive into the absolutely fascinating world of our restless genome.
So let's just start with this huge question. For a long, long time, we all thought the answer was a definite yes. But it turns out the reality is so much more dynamic and, honestly, a little bit chaotic. The agents of all this change are called mobile genetic elements, or MGEs. And their nickname, jumping genes, is, well, it's perfect. They are literally pieces of your DNA that can get up and move from one spot to another. And this visual right here shows you the core concept. You see that piece of DNA, the transposon? It literally hops out of its original spot and wedges itself in somewhere totally new. This whole process is called transposition. And get this, it's happening inside you, right now. So, here's the game plan for this explainer. We're going to explore this idea of our restless DNA, get to know these jumping genes a little better, and then we'll break down their two main ways of moving. After that, we'll see how our cells manage to keep them in check. And finally, we'll look at how they are a source of both incredible chaos and amazing creation in our evolution. Okay, so how do these genes actually jump? It's not totally random. There are actually two very distinct methods they use, and each one has really different consequences for our genome. The two main types are super easy to understand with a simple analogy. Think of class 2 elements as using a cut and paste function. The original piece moves, so the total number of copies stays the same. But class 1 elements, they use copy and paste. They make a duplicate of themselves, so the number of these things in our genome just keeps going up over time. This slide really helps visualize it. So over on the left, we've got that cut and paste method. You can see the element gets cut out and just moves to a new spot. See? No increase in the copy number. But look at the right. With copy and paste, the original one stays put while a brand new copy gets made and inserted somewhere else. That is the key difference. One just relocates, the other one proliferates. So how does cut and paste really work? Well, it's like a little three-step dance. First, an enzyme called transposase acts like a pair of molecular scissors and snips the element right out. Second, that same enzyme pastes the element into its new home. And finally, the cell's own repair crew rushes in to patch up the little gaps left behind. Pretty neat. Now, the copy and paste way of doing things is a bit more complex because it has this clever intermediate step. First, the original DNA is copied into a temporary messenger, an RNA molecule. Then, and this is the key part, that RNA message gets converted back into DNA. That's a process called reverse transcription. Finally, this fresh DNA copy gets stitched into the genome. Okay, so this all sounds pretty wild, right? I mean, if you've got pieces of your DNA constantly jumping around and making copies of themselves, what's to stop your entire genetic code from becoming a jumbled, unstable mess? And that is the crucial question. Because if this whole process runs wild, it can cause some seriously devastating damage to our DNA. So our cells are constantly performing this incredibly high-stakes balancing act, trying to allow for a little bit of change, without letting it all spiral into total chaos. This slide really shows you what's at stake here. On the left, you've got controlled movement, everything's stable. But on the right, uncontrolled transposition leads to breaks, rearrangements, and a super unstable genome. That's bad news. So our cells have evolved some very, very clever ways to keep a lid on all this. The first strategy is absolutely fascinating because it's all about controlling the scissors themselves. The cell can basically choose whether it wants to make a functional enzyme that can do the cutting or a broken one that just gets in the way. It's called alternative splicing, and it's a slick way to regulate transposition before it can even get started. And this is just brilliant. So in our regular body cells, the gene that codes for that transposase enzyme gets processed in a way that makes a shortened, totally non-functional version. This broken version actually gums up the works and represses any jumping. But in our germ cells, sperm and egg, the full active enzyme gets made, which allows for some evolutionary shuffling. All right, the second method is a bit more direct. The cell just makes specific proteins whose entire job is to slam the brakes on the whole transposition process. These repressor proteins have two main tactics. As you can see on the left, some of them just physically sit on the DNA like a roadblock, so the transposase enzyme can't even get on. Others, like on the right, just tackle the transposase enzyme directly, binding to it and shutting it down.
The third method is a form of what we call epigenetic control. It doesn't change the DNA sequence itself,
just how it gets used. Basically, cells can add these tiny chemical tags, called methyl groups, directly onto the DNA to silence these jumping genes.
And here you can see exactly how it works. These little methyl tags are like chemical do not touch signs. They physically block the transposase from getting a foothold. This also creates a very specific window of opportunity for these genes to jump, right after the DNA replicates, but before the new strand gets its own set of methyl tags. It's an absolutely beautiful bit of timing and control.
So after all this talk about regulation and damage control, you might be thinking these jumping genes are just a nuisance, right? Some kind of genetic parasite. But it's not that simple. They are a true double-edged sword.
On one hand, absolutely, they can be destructive. They can cause harmful mutations, mess up gene function, and in bacteria, they're famous for spreading antibiotic resistance genes around. But on the other hand, they are a massive engine of creation. Want a mind-blowing example? The evolution of the placenta in mammals. The very gene that lets the placenta fuse to the uterus came from a retrotransposon, an ancient jumping gene that our bodies co-opted to create one of the most important innovations in our entire evolutionary history.
So that's the crucial takeaway here. Even though they seem like selfish little bits of DNA just trying to make more copies of themselves, their actions are a fundamental driver of evolution. Without all this constant shuffling of the genetic deck, the incredible diversity of life on this planet, including us, would look very, very different.
Really, the big idea is that we need to totally update how we think about the genome. It's not some static, sacred blueprint. It's a dynamic, living document that is constantly being cut, copied, pasted, and rewritten over vast stretches of time.
And that kind of leaves us with a final big thought to chew on. If something that acts in its own selfish interest to just replicate itself is also a primary source of creativity and adaptation for the entire organism, what does that really tell us about the nature of life itself?